So, three types of solutions are iso, hypo and hypertonic. So, what does it mean? Iso means equal. So, here in isotonic, the external medium, external solution and the concentration of the cytoplasm of RBC, that is equal. In both it is equal. So if we put RBCs in isotonic medium, they will be as they were. They will not swell, they will not shrink. The water goes in and water comes out and they will be like the original one. So if isotonic solution is there, the concentration of solution outer and inner RBC, it is same. In hypo means less. Less means the outer solution is having lesser concentration than the cytoplasm of RBC. RBC has more concentrated cytoplasm than the outer solution. Outer is less concentrated. So what will happen? Now from the region of higher towards the lower, less concentrated, the water will go inside the RBCs and they swell up. As this is animal cell, it has no cell wall outside, they will burst. So when they absorb more water, they cannot hold this water and the RBCs, they burst in hypotonic solution. The third is hyper. Hyper means more. More means the outer solution is more concentrated than the RBCs. So here the solution, the concentration is more than the cytoplasm of RBC. So from the RBC, the water, it comes out in the outer medium. When the water comes out, they dehydrated or they shrink. So these are the shrunken RBCs in the hypertonic medium. So these are three: iso, hypo and hypertonic medium, and osmosis is dependent on the outer medium of the cell. So this is with animal cell. So these diffusion and osmosis, till we study now, these are naturally, they are occurring naturally. But sometimes, as in plants or in animal cell also, we see that sometimes potassium, calcium, these ions, or sodium, these ions, they go inside the cell. The cell needs these ions for its proper functioning. But what happens? Suppose this is the cell KKK, this is potassium ions, they are already inside the cell, which is in higher concentration than the outside medium. Here the potassium is less, inside the cell potassium is more, but even then more potassium is needed inside the cell. So it cannot go by the normal osmosis. So a special technique is there, that is known as active transport. What is active transport? Yesterday you have seen this inside the plasma membrane when we were studying the function, there are proteins. These proteins, they are having certain channels. So by this active transport, these potassium or ions of these can go in against the concentration gradient. What does it mean? It means although the concentration is more here, concentration is less here. So this should come out, but as the cell needs more from the lower concentration, from the region of lower concentration, these are going in towards the region of higher concentration. This is known as opposite or against the concentration gradient, and this takes place by use of energy, adenosine triphosphate or ATP. When ATP is used, then this active transport takes place inside the cell. So this is active transport. And in plants, in roots of the plants, some minerals, most of the minerals are being taken up by the roots. Even water is by simple osmosis, but ions and minerals which are dissolved in water, they are taken up by the roots of the plants by this active transport using the energy or ATP. So ions and minerals by the roots of transport, by the roots of plants, it is also being taken by active transport. So this is about the active transport. Now one more thing, unicellular organism which live in fresh water, they also absorb the oxygen from the water through this osmosis or diffusion. And second, one term is there, endocytosis. What is endocytosis? It is also related with plasma membrane, and endocytosis is taken up of food by amoeba or unicellular organism with the help of this plasma membrane. This is amoeba, a unicellular organism. When a food particle is present near it, this is nucleus, this is cytoplasm, and this is food particle. When amoeba cell, it sends food particles, it produces finger-like projections known as pseudopodia. And these pseudopodia, they encircle these food particles. How these pseudopodia are formed? They are the extension of plasma membrane. As plasma membrane is flexible, due to its flexibility, this extension or finger-like projections are formed known as pseudopodia. Next, these pseudopodia, they encircle this food particle and a food vacuole is formed. This food vacuole is inside the body of the amoeba. Now digestive juices, they digest this food and it is being absorbed by the cytoplasm of the amoeba. And this process is known as endocytosis, and this endocytosis is due to this plasma membrane. When the solid is taken, it is known as endocytosis, and when liquid is taken, it is known as pinocytosis. So these two terms, they are due to the presence of plasma membrane in unicellular organisms like amoeba. So this is about plasma membrane